Bismillahirrahmanirrahim. 5090 level biology to November 13. Today we're going to do a paper 1-1. This has some very interesting questions we need to address. We start with the quote, you can't go back and change the beginning, but you can start where you are and change the ending. And that's something for you all to think about. You haven't studied a lot, but from today onwards you can make a change in the way you are going to study and how you're going to take the exams. Question 1. A plant is grown in bright sunlight. The diagram shows what is seen when a cell from the leaf is placed under a microscope. After a few hours, a leaf from this plant is stained with iodine solution. What will be stained blue-black? Now we know it's the chloroplast where photosynthesis is going to take place and glucose is going to be made, and that is going to be converted to starch. Now we go on to question number 2. The diaphragm represents apparatus. The diagram represents apparatus used to investigate osmosis. So there's a tube and there's a solution level and there's a partially permeable membrane which is surrounding this tube here. This is the partially permeable membrane, and then there's water. So this is all water. And then inside this there is a concentrated starch solution. So this is the concentrated starch solution, which is of course going to be a little difficult for you to understand. Is starch cannot the large molecule cannot diffuse out of that partially permeable membrane. Which molecules will move across the partially permeable membrane and a change will occur? So naturally what you've got to understand is that the water molecules is going to enter, and then this is going to make this rise. So this solution level is going to rise. So water and rise was the answer to that. Now question number 3, the small intestine of a person contains a lower concentration of glucose than is present in the blood. The cells of the villi absorb glucose. By which process is the glucose absorbed? Everybody knows that is a syllabus point. And active transport against the concentration gradient. So it was active transport with the concentration gradient. That's a wrong definition. Diffusion was not there, so diffusion was wrong first, and then active transport. Only if you knew it, you knew this, you could do this. Then which statements are correct for all enzymes? Now the point was all enzymes, not just some enzymes. They're proteins, yes, all of them are proteins. They're secreted into the alimentary canal. No, enzymes are present in each and every cell of your body. Enzymes are present in each and every cell of a plant. I mean, I'm thinking of questions which could come from your MCQ exam. So they're secreted into the alimentary canal. No, that was wrong. They speed up chemical reactions. They speed up biochemical reactions. Yes, so that was correct. So 1 and 3 were correct. 4, none of them work at low pH. Well, that is wrong. Why is it wrong? Because they work at different pH. Some work at a lower pH, some work at a high pH. So it's all different, varies from enzyme to enzyme. Now coming on to question number 5, which signs show that a plant has been grown in soil deficient in magnesium ions? Now the word was magnesium ions. So you know magnesium ions result in less chlorophyll. So what happens in the leaves? In the leaves, the chlorophyll cannot be made. Just like if you don't take enough iron in your diet, then there will be less hemoglobin. Less hemoglobin means less oxygen carrying capacity. So yellow stem and yellow leaves, that was the answer to that. Then question 6, what is the function of each of these type of plant cells? Now, what is the function of each of these terms? So phloem cells and root hair cells. So phloem cells and root hair cells, phloems transport sugars. I mean, sugar is in a way a wrong word. Phloem transports sucrose. So sucrose is transported for the phloem, but I mean sugar they've allowed you to use, but you won't be able to use this in A levels. And then root hair cells are for what? Is for ion uptake. So root hair cells are for ion uptake, and the phloem is for sugar transport. 7. The diagram represents a cross-section of part of a leaf. How does the oxygen content of the air at X compare to normal atmospheric air when the leaf is in the light? So this was first was in the light and second was in the dark. Now air spaces, now these are air spaces. Now air spaces means what? What is happening here? Please everybody try to understand. What is happening here is that chloroplasts, these are the chloroplasts. So the chloroplasts photosynthesize in light and they're producing oxygen. So there will be more oxygen during the daytime. So more oxygen, more photosynthesis, more oxygen. But only photosynthesis takes place when there is light. So in the light, so more oxygen being released. So more oxygen and less oxygen. That was the point that you have to see, and the answer was D. Question 8. An investigation is carried out on digestion and absorption in the alimentary canal. The diagram shows the apparatus used. So this is actually we are trying to mimic or copy what is happening in the alimentary canal. So the diagram shows the apparatus used. The whisking tubing is permeable. Now this is the whisking tubing which I'm going to color in purple. So this is the whisking tubing, and they've said whisking tubing, they've underlined, they've given you that arrow. So the whisking tubing is permeable to small molecules such as glucose, but not large molecules such as starch. So starch cannot doesn't move out or move in, or it doesn't permeable nahi hai. After 1 hour, samples of water from the beaker, and inside this, what have we put in? Yes, another thing you've got to understand, this is a starch solution and amylase. So you've got to understand starch is going to be digested to maltose, and maltose is a reducing sugar. So look at the words that I'm using. Starch is digested to maltose. Maltose is a disaccharide, but it's a reducing sugar. After 1 hour, samples of water in the beaker are tested with Benedict's and with iodine solution. Which colors are obtained? Naturally, with Benedict's is going to be red, because red is the color if reducing sugars are present. And when we add iodine to this, the iodine will not change color. Iodine only changes color. Iodine only changes color when comes in contact with starch. But then you see, you've got to remember the starch molecules did not come out in the water. So iodine will not turn black. It will remain yellow-brown, because iodine's color is yellow-brown. So it will remain yellow-brown. Now, if you're not clear on this, you need to type in the Google search yellow iodine color and how it changes and what are the different colors which is available to you. Question 9. What are the substrate and end products of digestion by the enzyme lipase? Now, lipase substrate will be, so this is the enzyme. The enzyme is lipase. Now, what fits into it is a fat molecule. So this will be a fat molecule which is going to fit into it. And then what is it going to be broken down into? It's going to be broken down into fatty acids and glycerol. So the fat molecule is broken down into 3 fatty acid chains, fatty acids and glycerol. Now you can see this is what a fat molecule looks like. It's got a glycerol backbone, it's got 3 fatty acid chains. And actually what happens is that the bond between them breaks here. And so the bond between this breaks here, this breaks here, this breaks here. And what do we have? We have 3 fatty acids and we have the glycerol molecule all breaking apart. So the lipase breaks up the fat into the 3 fatty acids and glycerol. If somebody didn't know that, well, they need to know that for the exam on Monday. Then coming on to question number 10, food tests were carried out on 4 different substances. Which substances contain both protein and reducing sugar? For protein, everybody knows it's the biuret test. For reducing sugar, everybody knows it's the emulsion test. So the other two should be negative. Now that is what gives you the clue. Which one could be correct? You have to use it. You have to be very intelligent when you're doing. I always say it's not hard work, it's smart work. So when it is, if you knew that protein and reducing sugar, biuret and Benedict's, which is going to be positive, and emulsion test is for fats, and iodine test is for starch. So please remember that. This is how you would do this question. Then coming on to question number 11. In which direction do water molecules move in the phloem and in the xylem of a plant stem? Water molecules in the phloem can be moving in some phloem tubes up and in some phloem tubes down, not in the same one, but in different tubes. Because from some leaves it will be going to an upper area where there is a white flower or a red flower, and in another tube it will be flowing downwards towards the roots, because the roots are going to be also needing sucrose. In the xylem it is always up only. So xylem is up only and phloem is both up and down. Then coming on to question number 12. 4 similar leafy shoots are exposed to different conditions. The rates of water uptake and the rates of water loss are measured. The results are shown in the table. Which shoot is most likely to wilt? Now, what is wilting? Wilting is when the water uptake is less than the water loss. Like for instance, you don't drink enough water and you're sweating a lot. That's the same thing. Well, you won't wilt because you're a human being and you'll drink water, but a plant will wilt. If there is no water in the soil, it can't take up any water from anywhere else. So when the water uptake, I said, is less than the water loss. So the water lost has to be more. That's what you all have to see. Here the figure should have been less, and here the figure should have been more. In the all the other in C it is the same. Here the water uptake is 4 and the water loss is 2. Here the water uptake is 14 and 13. So that was the clue in it, that the water uptake figure should have been lesser, whatever the figure. I'm not saying 10 was necessary, could be 20, could be anything. But the water uptake figure should have been less. If it was 10, then it should have been 20, more water loss. So then, of course, the plant is going to become chungered and is going to wilt. Yeah, I love the word chunger. Question 13. What is the correct route for blood flow in a human? Right atrium to right ventricle, then the lungs, and then the left atrium and the left ventricle. Now, of course, if you are struggling with this, then you need to revise this chapter. You see, right atrium, then blood is going to go into the right ventricle, then it's going to go out to the lungs. So this is just asking if you know double circulation, and then it's going to return by the pulmonary vein, and this is going to be the left atrium, and then it's going to enter the left ventricle. So, I mean, it was very simple. Either you knew it or you didn't know it. If you didn't know it, well, mega chunga. Question 14. The table refers to blood vessels in the human body. Vessel, aorta, pulmonary vein, hepatic artery, hepatic portal vein, from to. So they've asked you what is, they left out certain things, P, Q, R, and S. Now, aorta from where to where? Now, you all know that the aorta arises from the left ventricle. So, only these two could have been correct. The rest was all rubbish. So, you narrow down to A and B. Then you read the question again and you say, okay, what was Q? Well, Q is pulmonary, vein, lungs, heart, oxygenated or deoxygenated. Well, you knew it was deoxygenated blood. So, pulmonary vein, sorry, is oxygenated blood. So pulmonary vein brings the oxygenated blood from the lungs to the left atrium. So you knew that was the only difference here. You can see there was this difference here. There's difference here as well. Left ventricle here also, like R. What was R? Hepatic artery, aorta, 2. So it could have been liver or kidney. You know, hepatic means hepatitis, means infection of the liver. So this was how you had to figure it out, and the answer was B, because S would have been oxygenated or deoxygenated. You know, hepatic portal vein is connecting the intestine to the liver. So when it's the blood is flowing, it's a vein. It's not, I wouldn't say it's always a vein, because pulmonary vein is also carries oxygenated blood. So this was going to the liver. Now, of course, all the oxygen would have been used up by the cells of the intestine. So this would be deoxygenated blood. So this is what you had to figure it out. I'm sorry, but you have to know these stuff. And if you didn't know it, well, you're not going to do inni, minni, mina, mo and say, okay, well, the answer is D. And then you high level changad, I think. Now, this is the most difficult question. I've been asked this many, many times by all of you on WhatsApp. The diagram shows an investigation of blood flow in the veins of the lower arm. Now, the most important thing in which you've got to look at is that veins, blood is flowing back to the heart. So this is the direction. If this is your arm, just draw a line on your arm and just draw a pencil with a line, draw with a pen, that this is the direction. So veins are carrying blood. So this would be the person. I'm sorry, I'm not very good at drawing human beings. So this is his arm, and the blood is going back and it will enter the vena cava and then it will enter the right atrium. So what do you see? Now, this was the most important thing. It said the diagram shows an investigation of blood flow in the veins of the lower arm. Now, a cloth is tightly wrapped around the arm at point Z. So if you tie this point, what is happening? The blood returning back is sort of stalling here a little bit, but it isn't good because it will go into the inner vessels, which these are the vessels which you can see on the top, but it will be diverted to the vessels inside the arm. So it's around Z and the veins stand out clearly. Why? Because there's sort of a little back pressure. Like if there's a blockage, there'll be, so that the veins stand out clearly. One finger, if you've ever given blood for some tests, if they take it from your elbow, or the area and the elbow, then they tie a sort of a tight band around your arm so that they can become very prominent, so that there's a little bit of stasis at the back and the veins become prominent. So one finger presses firmly on W. This is the point, W. Everybody clear? When another finger strokes the vein, as shown in the diagram. So there's another point here. So you just put your finger. Actually, if you were in class, I would have done it with you on my arms. My veins stand out very clearly, and I do this experiment to show it to you. When another finger strokes the vein as shown in the diagram, the vein lies flat. Means that usually this would be a thick vein, but now just there is a thin line. It lies flat. It should have been thicker, but now it just lies flat between points W and Y. So it just lies, it's like this. This is W and this is Y. Otherwise, it's thicker like this, W and Y. But why is it lying flat between points W and Y? Now let's read it again. 4 reasons are given to explain this, why the vein lies flat. The bandage at Z prevents backflow. It's not going to prevent backflow. It will just not, you see, the blood will just pile up here. It's not going to prevent backflow. It's going to circulate. So it will just not come back. So the bandage at Z doesn't prevent backflow of blood. Blood doesn't flow back in the veins. Then the finger pressed at W prevents more blood entering. Yes, this is what is happening. There won't be any blood entering from this side. Why? Because you're pressing on W. So 2 was correct. And a valve at Y. So there is a valve here at Y which closes. So blood doesn't go back. That is why it is lying flat. That is why it is lying flat. There is no blood entering it from either side. You see, it is this can't come back, so there is no back pressure. There is no back, so it prevents backflow of blood. So it's not that the valve just closes, so it doesn't allow it to come back. It is just a natural thing. It's not something which you're doing. So 2 and 3 were correct. But then why is 4 wrong? A valve at Z was the bandage area, prevents more blood from entering the vein. Now, that's absolute rubbish, because the veins at Z, what is happening? Let's look at it here. At Z, blood is flowing like this. This is the vein. So at Z, a valve at Z prevents more blood from entering the vein. No, the veins are in one direction. They're always like this. They just prevent backflow of blood. So even if there are veins at Z here, say this is Z, and there's a vein here, then you see it from entering. The vein is from this side. It just prevents the backflow of blood. Veins have blood entering from this side. So this was the main thing that you had to understand. Now this is very, very technically very important that you understand this question. Please take your time, listen to my answer at least twice, do this video again, just go back a bit and then listen to this whole story once again. So a valve at Z can't prevent more blood from entering the vein, because blood is entering from this side. And if the valve is even present, it just prevents backflow. So this was this very difficult question. Question 16, again another very difficult question. Students have asked a number of times. Seals are marine mammals. When they dive underwater, they are capable of respiring anaerobically for long periods. During this time, blood flows to the muscles is greatly reduced, but the muscles are able to tolerate high concentrations of lactic acid. The graph shows the concentrations of lactic acid and oxygen in the blood of a seal before, during, and after a dive. Take time to understand the question, please, everybody. So this is the period of the dive. So let's look at this point. The period of the dive is this area, and this is then the area before the dive, and this is the after the dive. So it says when the dive underwater, their capacity. So the graph shows the concentrations of lactic acid. So lactic acid is a certain a normal level, say this is 5. And then it stays the same during the dive, and then it rises. Where? Concentration in the blood in arbitrary units. And then let's look at the oxygen curve. The oxygen is, say, 20. So the oxygen remains 20 at all times before the dive. And then it just dives down, becomes less. And then it rises, and then it stays the same. And similarly case, you see the lactic acid rises and then it stays the same which was before as well. The same, maybe little bit, or maybe none. That's a zero. So what you need to understand is what explains the changes in the lactic acid concentration during time x, not during time x. What has happened to the lactic acid? The lactic acid has greatly increased. Let me give it another color. So the lactic acid has increased during time x and then it's decreasing. It's increasing and then decreasing during time x. This is time x. So it has increased in fact only. What explains the changes in lactic acid concentration during time x? Increased lactic acid production. Why is that wrong? Because they've stopped diving. The dive is finished. Increased rate of aerobic respiration can't remove the lactic acid. Reduced rate of anaerobic. Anaerobic has stopped. The dive is finished. Why is it? Because suddenly now there is the lactic acid from the muscle cells is entering the blood. It was being produced in the muscle cells, but now it is increased, it's entering the blood because it said in the question, the reduced blood flow to the muscles is greatly reduced. So as the muscle cells can tolerate high levels of lactic acid, now suddenly there's increased blood flow to the muscles, and that is going to result in the lactic acid increasing. Technically difficult question, totally agree with you. Then coming on to question number 17, which process does not result in an overall loss of energy from the organism? Loss of energy, a boy running 100 meters, muscle contraction, energy loss. Respiration in animals, yeah, energy is released, and the energy is going to be released and going to be the germination of seed, lots of energy being used up. So overall loss, so you have to think of it, which one is not a use of energy. Photosynthesis is in a green plant does not use energy. It converts light energy to chemical energy. You have to know this. You have to be very, very good in biology to understand this. That it was actually a question on the uses of energy in the human body and in plants. So photosynthesis is a process which does not use energy. And question 18, a person begins to smoke a cigarette at time X. The graph shows how their heart rate changes. So heart rate increases when the person is smoking, and then it sort of decreases. Which substance in cigarette smoke is the main cause of the change in heart rate between 10 and 18 minutes? When nicotine results in increasing the heart, it has a direct effect. And it also causes vasoconstriction, and that, of course, increases blood pressure. So that is something factual which you have to know. Then 19. In a kidney dialysis machine, which substance cannot diffuse through the dialysis membrane? Very, very easy. It had to be either a protein. Now, they didn't give you a protein because this is what is written in the syllabus, protein molecules. But then when they gave you insulin, it's only the very smart students. It's only the very, very, I wouldn't say clever, I wouldn't say intelligent, I would say hardworking students who have revised their biology and who know that insulin is a protein molecule. The rest of it was all glucose and sodium and urea was all rubbish because actually they said cannot diffuse. We want urea to move out of it so that we want to remove the urea. That's the function of the dialysis machine, that the kidneys are not working, so we want to remove the urea. Question 20, the diagram shows some of the structures seen in a section through the human skin. What is the function of structure X? And what are the X? X are receptors to detect changes in temperature. You enter this room is very hot, and you say, oh my God, this room is very hot. Well, how did you come to know? You didn't see it through your eyes or your ears or your nose. There's receptors in your skin which can detect this. And this, of course, please remember another question. I would give you this question. This is what? This is the sensory neuron. This is the sensory neuron. And if I had to ask you a question on the motor neuron, then I would give you something as sweat glands here, and I would say, what is this nerve going towards it? And this would be a motor neuron. So good question. So what is the function of structure X? Sensory neuron, these are receptors in the skin which detect changes in environment temperature. You touch something hot and you say, oh my God, that's very hot. Well, how did you come to know? Well, it's because in your skin you have temperature receptors. Last question, 21. Which row describes the shoulder joint of an arm? The shoulder joint is between the two bones, scapula and humerus. So you only had to figure out, was it this or was it this? Then, of course, if you knew this was a ball and socket joint, and the fact that the action can be rotation. But if I had asked you, if I changed this and I said which one is the elbow joint, then what answer would you give me? The answer would be no, none of these I think would fit in here. It's the humerus, radius, humerus and ulna. There's none there. It's a hinge joint and flexion and extension. So please remember this and wish you all the best. And thank you very much for watching the videos, and please do give likes so that and do subscribe to the channel. Thank you very much.